Okay, so let's take a look at the possible blood types that we could have. So the blood types are A positive, B positive, O positive, AB positive, A negative, B negative, O negative, and AB negative. Notice that in order to properly name the blood type, you need both the letter and positive or negative. So there's no such thing as just type A blood, type B blood. It's always followed by positive or negative. Okay, so here's this little chart to help us reinforce our understanding of antigens. So let's look at A positive blood. Okay, so this red blood cell, I've modified it. Of course, our red blood cells are these biconcave little discs, but I just did that like that to make it easier. So A positive blood, a person with A positive blood will have A antigens on their red blood cell, okay? But this person is positive. And when we talk about positive or negative, we're really discussing the presence of an Rh antigen. So this is just an additional antigen. Different textbooks are going to call it different things. We're going to call it the Rh antigen, okay? So if you have positive blood, you are going to have an Rh antigen. Okay, let's compare this to A negative blood. These people will only have the A antigens. They're negative, they do not have Rh. Okay, let's go to B positive. So B positive blood, B antigens, and the Rh factor. B negative blood, just B antigens. AB positive blood. These individuals will have A antigen, B antigen, and Rh antigen. AB negative, just A's and B's. Okay, O positive blood. These individuals will only have the Rh antigen. O negative blood, they have no antigens that we're going to talk about on the red blood cell. Okay, so no A's, no B's, no Rh's. Okay, so when you're thinking about blood typing, have these pictures in your head. Okay, what does the red blood cell actually look like? So when we think about the blood typing cards that we did in lab, that was on your practical and your quiz. So we have this card and it has three basically little wells. Okay, this well right here, we say it's anti-A. This guy is anti-B. And this guy is where we place anti-D. Okay, so on the blood typing card in class, this said anti-D. For all intents and purposes, you can think of this as anti-Rh. It is the same thing. It's just a different way to call it. Okay, so what these really are, these are antibodies. Okay, so when you think about those three serums that we had, right? You had a blue serum, a yellow serum, and a clear serum. Those were antibodies. Okay, so they are little tiny proteins that we are putting onto the wells. Okay, so the first thing that you did when you did this experiment is you put your own blood on each one of these wells. Okay, and this was assuming that we do not know your blood type. So we put a drop of your blood on each one of the wells. The next thing that we did is we put antibody A on one of the wells. So here's antibody A. Okay. Now, if you remember from our definition of what an antibody is versus what an antigen is, recall that an antibody binds an antigen. But remember, it will only bind its specific antigen, okay? So when you put antibody A onto this blood, what happens, you mix it and you just see a really nice and smooth reaction, okay? Now let's do antibody B. This time, let's do a different color here. This time you put a drop of antibody B. And now, when you mix that with your blood, instead of seeing something really nice and smooth, you see this kind of clumping. Okay, the blood got speckly. Then, when you put antibody D or antibody RH, right? It's the same thing. You also saw speckling. Okay, so the first thing that we need to talk about is what does the speckling really mean? So the speckling that's happening here and that's happening here, this is called agglutination. It is not called coagulation. It is not called clotting. Okay, it's a very specific process called agglutination. And what agglutination really is, and here's the key to blood typing, is evidence that the antibody found its antigen. Okay, that is the key. If you see clumping, if you see this agglutination, it's evidence that the antibody that you put has found its antigen. Okay, so example, antibody B. This is a little protein that is roaming around looking for B antigens. And if it finds B antigens, it's going to bind to that B, and that's what is causing the clumping. Okay? So when we think about this, let's go to antibody A. Antibody A, we put antibodies searching for A antigens onto the blood, and there's no agglutination, there's no clumping. So we can infer that the antibody did not find its antigen. So there were no A antigens present on the red blood cells. Right? So let's go over here and look at our chart. So if we can't have anything with A's, we know we can't have A positive, A negative. We couldn't have AB positive, and we couldn't have A negative. So we know our blood type has to be B positive, B negative, or O positive, O negative. Okay? Let's go over here to antibody B. When we put an antibody searching for antigen B, we saw clumping. So we say, yes, there are B antigens on the red blood cell. So we go over here, well, we know that it can't be O positive blood, right? Because O positive blood doesn't have any B antigens. We know it can't be O negative blood. Okay, there's no B antigens on that red blood cell. So we're left with, it's either B positive or B negative blood. And that's where this comes in. This last antibody is where we say positive or negative. So we see clumping when we treated it with an antibody against RH. So that means is that this little antibody was roaming around looking for RH antigens, and it found an RH antigen and it bound. For that reason, we say that yes, there were RH antigens on the red blood cell. And then we look at our chart. Okay, who had RH antigens? Not B negative. So the answer had to be B positive blood. Letter, positive or negative, makes the complete blood type, B positive blood.